the digestion of food is actually divided into two processes. One is the mechanical process and the other one is the chemical process. Starch. So where was starch present? Starch is nothing but a form of carbohydrate, complex carbohydrate that is present in the food. So always remember, if the HCl has to convert the inactive pepsinogen into active pepsin, then its pH should be 1.8. Then only it can convert. Hello everyone, a warm welcome to another session on chapter 16, that is Digestion and Absorption. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the different organs that are present under the digestive system or the alimentary canal. So once we understood that, in today's session, we shall move into studying about the process that occurs in digestion, that is how exactly the food gets digested when it travels through the alimentary canal. So to begin with, we shall study the digestion of food. So the digestion of food is actually divided into two processes. One is the mechanical process and the other one is the chemical process. So you can guess when I tell about mechanical process, right? Something involving with the biting, chewing, everything is the mechanical process. And where does this mechanical process occur? In our oral cavity or the buccal cavity, right? So we shall start studying with the mechanical process, wherein the first process of digestion is wherein we ingest the food or take in the food into our mouth, and then in the mouth using our teeth, we chew it properly, wherein the food mixes with the salivary enzymes and all that, and what happens we shall see. So talking about the mechanical process. So in this mechanical process, the buccal cavity or the oral cavity that is present in the mouth actually involves in the mechanical process. So the buccal cavity performs two major functions. One is mastication of food. So mastication of food, the chewing of food in our mouth is called as the mastication of food, making into smaller bits or cutting the food into smaller portions, and facilitation of swallowing. Next, after mastication, the food would have mixed with the saliva and all that, and it would have become very, very soft and easy to swallow. Then we swallow it, right? So therefore, there are two processes of function involved in the mechanical process that occurs in the buccal cavity. One is the mastication of food will occur, and the second one is the swallowing of the food, masticated food will take place. So the teeth and the tongue with the help of saliva actually helps in the mastication. So mastication, so chewing. So of course to chew we need our teeth, right? But is the teeth alone enough? No. While we are chewing, we can feel our tongue moving. The tongue actually tilts the food. Just like how a large tank will be there to mix the cement and the jelly that is there, right? Jelly stones. So just like that, in our mouth also there is this tongue which helps in the circulation or movement of food inside our mouth as and when we chew it. So therefore, the teeth and the tongue are the two organs that are present in the buccal cavity that helps in the mastication of food. And as and when we chew, the salivary glands get activated and the salivary amylase will be produced, and this salivary amylase will moisten the food and it will mix up the food thoroughly. And the mucus and the saliva that is secreted during the mastication, it actually helps in moistening or it helps in lubricating the food and also helps in adhering the food. So we always, when we swallow the food once it is masticated, it goes completely inside, right? It will not go bit by bit. Why? Because all these masticated food particles are held together by the saliva. So that is the reason. So the masticated food, now it is called as the bolus. So the masticated food that is there in the form of a paste. Just imagine a mixie, that is a grinder, right? First you add all the ingredients to it, which the ingredients looks bigger in size. Then you switch on the mixie, the blades will just make it into smaller bits. Once it becomes into smaller bit, then you'll add a little bit of water, right? And after you add water, it will become like a paste. Now that is the bolus. Right, so just like that also in our mouth, exactly like how in a mixie the function occurs or in a grinder, same way inside our mouth also the function occurs, and that paste like food that is formed is called as the bolus. And that bolus is then conveyed into, or it is pushed into the pharynx and then into the esophagus, that is the food pipe, and then the swallowing or the deglutition. This is called as the swallowing or the deglutition process. So first is mastication. After mastication, deglutition or swallowing will occur. So this is the mechanical process that occurs during the digestion process. So first is ingestion. So ingestion occurs in the mouth. Ingestion means taking in of the food into the mouth. After the ingestion, here from the mouth, oral cavity up till the esophagus, it is the mechanical process. And next, after that, once the food enters into the stomach, then it enters into the chemical process of digestion. So this was about the mechanical process that we studied. So next, what happens to the bolus? Okay, so the bolus has been swallowed. It has entered into the esophagus. First it enters into the pharynx and then it enters into the esophagus. After it enters into the esophagus, it further passes through the esophagus by successive waves of muscular movements that occur in the esophagus. So the esophagus has to push down the food, right? If the food has to be pushed down, a muscular wave-like function or contraction occurs in the esophagus, which is called as peristalsis. So because of this muscular wave-like contractions that occur in the esophagus, which is called as peristalsis, the food easily moves down to the esophagus. Because what happens here is when we are swallowing, we put a pressure. That pressure will allow the food to move into the pharynx. But we cannot put a voluntary pressure, cannot be put when the food enters into the esophagus. So therein, the esophagus has such a mechanism wherein it will undergo a wave-like contraction process, which is called as peristalsis. And this peristalsis is the process that helps in the movement of food down through the esophagus. Now, the gastroesophageal spinster. So here, between the esophagus and the stomach, there is a flap that covers the esophagus and the stomach, which is called as the gastroesophageal spinster. So this gastroesophageal spinster, it controls the passage of the food into the stomach. As and when we are, so it will not be open all the time. What happens if it is open all the time? Whatever food enters into the stomach, it will go back or it will revert to the esophagus. That should not happen, right? So that is why between the mouth of the esophagus at the bottom and the stomach, there is a small flap-like structure or a covering that is present, which is called as the gastroesophageal spinster. When we chew the food, when the food enters, becomes a bolus, that bolus enters into the pharynx. From the pharynx, the bolus enters through the esophagus where it travels down through the esophagus by a wave-like contraction, which is called as peristalsis movement. And then once it reaches the gastroesophageal spinster, now it has to enter into the stomach. So the gastroesophageal spinster will open, it will allow the food to enter. Once the food passes, very soon it will close. Why? Otherwise, until the next time we food, what might happen? The food particles might just go up back into our mouth. It might rise through the esophagus. So to prevent that, this gastroesophageal spinster actually controls the passage of the food into the stomach from the esophagus. And the saliva secreted into the oral cavity, it contains a lot of electrolytes and enzyme. So one of the best example of an enzyme that is secreted by the salivary glands is the salivary amylase and the lysozyme. So these salivary amylase and lysozyme also helps in the partial digestion of the carbohydrates. So carbohydrates, if it has to get partially digested, then this salivary amylase should be there. So therefore, along with the mechanical digestion in the mouth itself, the chemical digestion would have also started. Why? There in the mouth, there is secretion of the salivary amylase and the lysozyme, in which the salivary amylase is one such enzyme that starts the carbohydrate digestion in the mouth. So that is the reason. So this was about the mechanical digestion process. So next, moving on to the chemical digestion. So I told you chemical digestion begins in the mouth itself, not just in the stomach, but a partial digestion of a particular biomacromolecule would have begin in the mouth itself. So what are the biomacromolecules? The lipids, carbohydrates, the proteins. So whatever food we eat, do you think it contains only carbohydrates, or it will contain only vitamins, or only lipids, or only proteins? No, it is made up of a mixture of all the biomacromolecules such as the lipids, the carbohydrates and the proteins. So each of these biomacromolecules, once it reaches the small intestine, it should get completely digested, right? So therefore, each of the process, that is in each part of the alimentary canal, exactly what types of chemical digestion will occur, we shall study. So the chemical process. So first we'll talk about the mouth. So in the mouth, what has happened? The bolus has formed, right? So bolus, that is the masticated food which has mixed with the saliva and all that, and it is formed a paste-like form which is called as the bolus. Now, this chemical process of digestion in the mouth is initiated in the oral cavity, or it begins in the oral cavity, that is in the mouth, by the hydrolytic action of carbohydrate. So remember, there is a particular enzyme, that is salivary amylase, which is secreted by the salivary glands that are present. So we have the submandibular gland and all that, right? So these glands actually secrete the salivary amylase. So these salivary amylase are one such enzymes that are capable of breaking the bonds between the carbohydrate molecules. So they are also called as the carbohydrate splitting enzymes. So these salivary amylase, they work on the carbohydrates breaking down. So don't think that complete digestion of carbohydrate would have occurred in the stomach. Only partial digestion. Only the bonds between the carbohydrate will be broken down by making it into simpler forms by the salivary amylase. So that is the first process that occurs. So in simple way, you can remember that the first process of digestion, chemical digestion, begins in the mouth, wherein the salivary amylase acts on the carbohydrates, breaking them into simpler forms. So now in the mouth itself, whatever food we have eaten, in that food carbohydrates will get broken. So about 30% of the starch is hydrolyzed in the mouth itself. Why? In the mouth, the salivary glands secrete the enzyme salivary amylase, which has an optimum pH of 6.8, and this 6.8 pH carrying salivary amylase is capable of breaking down about 30% of the carbohydrate or the starch that is present in the food into simpler forms. That is, it breaks it into a disaccharide, for example, maltose. So here they are complex polysaccharide forms. Those polysaccharides will be broken down into simpler disaccharide forms. Now further, those disaccharides have to be broken down into monosaccharides and then broken down into even more simpler forms and made available to the body. That process and all will occur as the food enters into the stomach, small intestine and all that. But 30% of the digestion of the carbohydrate or the starch will occur in the mouth itself with the help of the salivary amylase enzyme that is having a pH of 6.8, wherein the salivary amylase will break the complex carbohydrates into disaccharide forms of carbohydrates, which is maltose. So this, and also understood in the previous slide, that there is not just salivary amylase that is secreted in the oral cavity. So in the oral cavity, there is also secretion of the enzyme called as lysozyme. So the lysozyme that is present in the saliva, its main function is to provide protection to our body. So we tend to eat a lot of raw food, right? Whoever is in a diet, they prefer eating raw vegetables. And also, who doesn't love to eat fruits, right? Do we cook the fruit and eat? No, we eat it raw. So do you think it is in a pure form without any microbes or anything? No. Raw food will always have microbes in it. So imagine what would have happened if you had eaten an apple. They would have, of course, there would have been some microbes which is not seen to a naked eye, right? We, without knowing, will just eat it off. Every now and then we would have fallen sick. But actually, we need to thank this enzyme which is called as the lysozyme. So this lysozyme is one such enzyme that is secreted in the oral cavity, or it is present in the saliva, that actually has some antibacterial property. So therefore, any bacteria that is there in the raw food, it will kill it. So therefore, our body will not get affected and we will be free of infection. So that is the function of the lysozyme. That is, it is an antibacterial agent that prevents the infection. So what happens? We have represented it in a simple equation here. Starch. So where was starch present? Starch is nothing but a form of carbohydrate, complex carbohydrate that is present in the food. So that starch that is there, it will mix with the salivary amylase or saliva, and that saliva contains salivary amylase which is having an optimum pH of 6.8, and these salivary amylase, they will break down the starch into simpler forms, that is maltose, which is a disaccharide. So it is a disaccharide now. So the complete breakdown of carbohydrate has not occurred here. Further breakdown will occur. We shall study about it later. So this was about the chemical process of digestion that occurred in the mouth. Now we have the food has become a bolus. That bolus has entered into the pharynx, and from the pharynx it has entered into the esophagus. From the esophagus it enters into the stomach. Esophagus does not produce any enzyme. So therefore, esophagus doesn't help in the process of digestion. So directly once the chemical digestion occurs in the mouth, then the next step where the chemical digestion occurs is in the stomach. So we shall see the chemical digestion process in the stomach. So the chemical digestion process in the stomach. So the mucosa that is present in the stomach has gastric glands. So mucosa is a layer of the stomach, and this mucosa has gastric glands present in them. And these gastric glands have three major types of cells. That is, they have the mucous neck cells, they have the peptic or chief cells, and they are made up of the parietal or oxyntic cells. So what are the functions of these? We shall see. So the stomach is covered by a membrane called as the mucosa. That mucosa has certain gastric glands being present, and these gastric glands are made up of three types of cells. One is the mucous neck cells. The main function of the mucous neck cell is to secrete mucus. Next one is the peptic or the chief cells. So their function is to secrete the proenzyme pepsinogen. So the third one is the parietal or oxyntic cells. Their function is to secrete HCl and certain intrinsic factors that are very, very important for the absorption of vitamin B12. So vitamin B12 is one such vitamin that is not produced in a body. We can only get it in the form of a supplement or by a food. So food, usually we cannot get enough vitamin B12. That is why we prefer usually taking a supplement to fulfill our body's vitamin B12 content. So whatever vitamin B12 has been taken in the form of food or through a supplement, that has to be absorbed by the body. If it has to be absorbed by the body, then this particular parietal or oxyntic cells will help. So that is a function. So now, what does the stomach? Now we understood. Therefore, because of the presence of the different types of cells in the salivary glands, what are the different enzymes or chemicals that are secreted in the stomach that helps in the process of digestion? One is the mucus, the other one is the enzyme pepsinogen, and the third one is the HCl. So all these are the chemicals that are secreted by the stomach, therefore helping in the process of digestion. Where and when these enzymes or chemicals participate in the process of digestion, we shall see. So the stomach actually stores the food for about 4 to 5 hours. That is why after 4 to 5 hours, you might have noticed your stomach will start to grumble, right? It will start to grumble and you'll feel like as if there are butterflies or something running inside your stomach. It is because after 4 to 5 hours, the stomach would have been empty because the digestion process would have got completed, and the enzymes will be working on to search for food to go into the next action of digestion. So that is why we hear all sorts of noises, the sounds and all that in our stomach after 4 or 5 hours. So that is why every 4 or 5 hours we need to feed something into a stomach. So the food mixes thoroughly. So now the bolus that is there has entered into the stomach. In the mouth, the partial digestion of the carbohydrate had already taken place, and now that food has entered into the stomach. In the stomach, the gastric juices that are there, be it the HCl, the pepsinogen or the mucus, they have time, that is 4 to 5 hours, to complete the digestion process. So the food that has entered into the stomach, it thoroughly mixes with the acidic gastric juice that are present in the stomach, and these acidic gastric juice will further churn or it will further smoothen the food particles, or it will result in churning movement of the food particles, and it is called as chyme. Now, once the food reaches the stomach, what happens? The food mixes thoroughly with acidic gastric juices. After the food mixes thoroughly with the acidic gastric juices, the complete food should be covered with the gastric juices. Therefore, the churning process that is there, or the mixing process that is there, will occur, and that particular food which is mixed with the acidic gastric juices is called as the chyme. So, bolus entered into the stomach. That bolus got converted into a chyme by mixing with the acidic gastric juices. So, that is how it occurs. So, the mucus, talking about the mucus. So, the mucus cells that are one of the components of the mucosal layer of the stomach, that they actually produce mucus and bicarbonates. So, the mucus and bicarbonates that are present in the gastric juice, they play a very, very important role in lubrication, that is in moistening the food and also in protecting the mucosal epithelium or the mucosal covering of the stomach from the highly concentrated hydrochloric acid. So, what is there in the stomach? Stomach secretes pepsinogen, it secretes mucus, it also secretes HCl. HCl is concentrated in the stomach, and that HCl can harm the mucosal layer of the stomach or the mucosal epithelium of the stomach. So, that is the time when the mucus and the bicarbonates come to help. So, the mucus and the bicarbonates that are there in the gastric juices of the stomach, they actually help in lubrication of the food, and not just that, they also help in protecting the epithelial lining or the mucous epithelial lining of the stomach from the harmful effect that can be caused by the concentrated hydrochloric acid that is present in the stomach. So, next, talking about these other enzymes. So, we understood what does the mucus do? Mucus actually helps in lubricating the food and all that. Now, that lubricated food has to be broken down again into simpler forms, right? Carbohydrate digestion, still carbohydrate has to be digested, still proteins has to be digested, fats have to be digested. Carbohydrate digestions partially had already occurred in the mouth. What about the others? Right? They should get digested. So, this food that is being lubricated by the mucus and the bicarbonates, they are acted by the pepsinogen. So, the pepsinogen is one such enzyme. In the presence of HCl that is there in the stomach, it actually converts the pepsinogen. So, pepsinogen is actually inactive form. So, when the food enters, when the bolus enters into the stomach, that is the time when the pepsinogen becomes active. How does it become active? It becomes active by taking the help of, by combining with HCl and forming an active compound or an active enzyme called as pepsin. So, pepsinogen is inactive, pepsin is active. So, this pepsin, now pepsin has become active. What is the function of the pepsin? In the food that we have taken that has entered into the stomach, proteins are there, right? So, that proteins will be broken down into proteoses and peptones by the pepsin. So now what all digestion has already occurred? Partial carbohydrate digestion took place in the mouth. When it reached the stomach, in the food, proteins that are there, those also got digested into proteases and peptones. So much of digestion has occurred. So small amounts of lipases, so the gastric glands that are present in the stomach, they also secrete some other enzyme called as lipases. And apart from that, the HCl that is secreted by the stomach, it provides an acidic pH of 1.8, which is optimal for pepsin. So always remember, if the HCl has to convert the inactive pepsinogen into active pepsin, then its pH should be 1.8. Then only it can convert. So this was about pepsin. That is how pepsin was made. Like pepsin is nothing but an active form of the pepsinogen. So pepsinogen is inactive. That inactive pepsinogen should be made active. How? It is made active by the HCl, which is having a pH of 1.8. Now when the pepsin becomes active, the protein that is present in the food will be broken down into proteases and peptones. So this is how the protein digestion begins in the stomach. Carbohydrate digestion begins in the mouth, protein digestion begins in the stomach. And there is also one more important enzyme that is produced, which is called as renin. So renin is usually not produced in adults. Renins are usually produced in the babies who are still dependent on the mother's milk. Why? Because the mother's milk contains proteins which cannot be easily digested by the pepsin. So that is why in babies, they have a particular enzyme being produced in the stomach, which is called as renin. So this renin is a proteolytic enzyme that is found in the gastric juice of infants or babies, and they help in the digestion of the milk protein. So mother's milk, it contains a lot of proteins. If that has to be broken down into simpler forms, then the infants, they should have renin that are produced in the gastric juices of the stomach. So that is about renin. So renin, usually adults will not have renin. So this was about the digestion of food in the stomach. So moving further, what else happens in the stomach? What other digestion occurs? We shall see. So various types of movements are generated by the mucosal layer of the small intestine. So next, after the food enters into the stomach, there the carbohydrate would have got partially digested. The digestion of proteins also occurred, wherein the proteins in the presence of pepsin got converted into peptones. And next, these will enter into the intestine. So now in the small intestine, what happens? Different types of movements are generated by the muscularis layer that are present in the small intestine. So these movements help in thorough mixing up of the food. Like how the food in the stomach, it became a chyme, right? Because of thorough mixing. Just like that in the small intestine, the intestinal wall has a muscularis layer that actually helps in the proper mixing of the food. So these movements help in thorough mixing up of the food with various secretions that or the enzymes that are secreted by the intestine, therefore facilitating further digestion. So now the food, after completing its digestion in the stomach, it enters into the intestine. Small intestine is the region where the complete digestion of the food will take place, and next it will enter into the large intestine. So here talking about the small intestine. So the bile, the pancreatic juice and the intestinal juice are the secretions that are released into the small intestine. So in the small intestine, what are the secretions that are released? One is the pancreatic juice. The small intestine will not directly produce certain enzymes like that of the stomach. So stomach, it had mucosal layers which actually produced its own enzymes. That is, it produced the pepsinogen, it produced HCl, and it also produced mucus, right? But intestine cannot produce most of the enzymes. So it collects the enzymes from other organs. So say for example, bile. Bile is secreted by the liver, it is stored in the gallbladder, and from the gallbladder it is secreted into the intestine. Next, talking about the pancreatic juice. Pancreatic juice is produced by the pancreas and then it is supplied to the intestine. And the intestinal juice, obviously only that is secreted by the intestine. Other secretions that are released into the small intestine. So the pancreatic juice and the bile juice are released through the hepatopancreatic ducts that are there. So if you can see here, this is the small intestine. Into the small intestine from the pancreas and from the liver, actually these juices enter into the small intestine. That is the bile juice and the pancreatic juice enters. And the pancreatic juice contains a lot of inactive enzymes such as trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases and nucleases. These are all inactive enzymes. They have to become active and then they have to start the process of digestion. So what are the different inactive enzymes secreted by the pancreatic juice? They are the trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases and nucleases. So we shall see what happens next. So how do each of these enzymes get activated and what role they play in the process of digestion in the small intestine, we shall look into. So trypsinogen is activated by an enzyme called as enterokinase. So trypsinogen is activated. So which produces trypsinogen? Pancreatic juice, right? Pancreas will secrete pancreatic juice. These pancreatic juice contain a lot of inactive enzymes. These inactive enzymes, when it reaches through the small intestine, it has to become active, right? How will it become active? So the enzymes to make these inactive enzymes active are present in the small intestine. So in the small intestine, there is one enzyme present called as the enterokinase. So this enterokinase will act one of the enzyme that is present in the pancreatic juice, that is called as the trypsinogen, and this enterokinase is actually secreted by the intestinal mucosa of the enzyme and they convert the trypsinogen into active trypsin. So trypsinogen came into the small intestine from the pancreas. It is an inactive enzyme. Once it reached the small intestine, that trypsinogen got converted into an active form called trypsin in the presence of an enzyme in the intestine called as the enterokinase. So this bile actually helps in the emulsification of fat. So what is emulsification of fat? It is nothing but the breaking down of fats into very small simple components. So bile, apart from that, it also activates the lipases enzyme. So lipases enzyme was one of the inactive enzymes present in the pancreatic juice. If the inactive enzyme that is present in the pancreatic juice, that is lipases, if it has to get activated, then bile is important. So remember, understand, bile does not produce any enzymes. So we had studied about the pancreatic juice. In the pancreatic juice, it had various enzymes, right? That is trypsinogen, lipases, then nucleases, various forms. But they were all in the inactive form, where trypsinogen became into active form when it was released into the small intestine, and in the small intestine enterokinase enzyme was there which actually activated the trypsinogen, converting it into trypsin, right? And that trypsin will act on breaking down the food into even smaller, simpler forms. Next is bile. So bile, I told you, it does not contain any enzymes. What it contains? It only contains some pigments such as bilirubin and bilirubirin. That is why bile is usually somewhat yellowish in color, because of the presence of these pigments. And not just that, they contain bile salts, they contain cholesterol, phospholipids, all these are there, but enzymes are not there. So if enzymes are not there, what is the function of the bile? Bile has a particular function wherein it helps in breaking down the fats, and not just that, it also helps in activating the lipase enzyme that was secreted by the pancreatic juice. So next here, so what happens in the small intestine exactly, we shall see. Now the food has entered into the small intestine, the pancreas has secreted the pancreatic juice, the bile has secreted the bile, and the bile helped in the activation of lipases. In the pancreatic juice, also trypsinogen was present. That trypsinogen was activated by the enzyme enterokinases that was present in the small intestine. So much we came to know. Two enzymes got activated. What are the two enzymes? One was lipases got activated and other one is trypsinogen got activated into trypsin form. So now what happens exactly in the small intestine? We shall see. So the secretions in the small intestine, there are some special cells called as the brush border cells. So the brush border cells that are present on the mucosal lining of the small intestine, along with the secretion of the goblet cells that are present in the small intestine, they constitute the intestinal juice, which is called as the succus entericus. So intestinal juice is also called as the succus entericus. So these succus entericus are nothing but secretions of the brush border cells of the mucosa and also the secretions of the goblet cells that are present in the intestine. So altogether the secretions of the brush border cells of mucosa and the goblet cells together, it is called as the intestinal juice, or it is also called as the succus entericus. This is one very important one mark question. So what is succus entericus? They can ask in the exam. So this juice, what is the juice? It is succus entericus. So this juice contains a variety of enzymes like disaccharides. Example, it contains maltase. Maltase is important. Why? Remember, in the mouth, digestion of carbohydrate had begun, wherein that digestion of carbohydrate, the complex carbohydrate got broken down into a disaccharide called maltose. It got broken down into a disaccharide. Now it has to get broken down into a monosaccharide, right? So if maltose has to be broken down into a monosaccharide, maltase is important. So that maltase gets secreted now by the succus entericus. It contains actually the maltase, and it contains dipeptidases, it contains lipases, it contains nucleosidases, all these. So the mucus along with the bicarbonates from the pancreas, it protects the intestinal mucosa from the acid, as well as it provides an alkaline medium, that is, it maintains the medium of the alkaline nature of the small intestine with a pH of 7.8 for various enzymatic activities to occur in the small intestine. So what exactly we shall see? So you can see the mucus cells, mucus layer. So the mucus cells secrete some juices, and also the goblet cells that are there, these also secrete some juices, and the juices that are secreted by the mucus cells and the goblet cells together they form the succus entericus. So we shall see next what happens in the small intestine. So now proteins are there. Some of the proteins got broken down in the stomach itself into proteoses and peptones, right? So now in the intestine, what all are there? Proteoses is there, proteins is there, peptones is there, maltose is there. Maltose breakdown occurred in the mouth, right? During the carbohydrate breakdown in the mouth. So in the intestine, all this is there. So the chyme that was present in the stomach, it entered into the intestine. So the chyme reaching the intestine are acted upon by various proteolytic enzymes of the pancreatic juice, and how the reaction occurs we shall see. So here proteins are there. So in that chyme what is there? Proteins. Because did all the proteins get broken down? No. Only few of the proteins got broken down into peptones and proteoses. So remaining proteins are there in the food in the small intestine. Peptones are also there, proteoses also there. They have to be again further broken down. So proteins, peptones and proteoses in the presence of trypsin. How did we get trypsin? Pancreas secreted pancreatic juice which contained trypsinogen. That trypsinogen was in the inactive form. Once it reached the intestine, in the intestine, enterokinase enzyme got secreted. That enterokinase enzyme converted the inactive trypsinogen into active trypsin form. So that is how it got converted. Now that active trypsin, which is also called as the chymotrypsin, along with the presence of the carboxypeptidases, it breaks down the proteins, peptones and the proteoses into dipeptides. So this is one reaction that occurs in the small intestine. So proteins, peptones and proteoses got broken down into dipeptides. So because we know that proteins and all that are polypeptides, right? They got, imagine from long chains of amino acid, they just got broken down into dipeptides. Next, fats. Fats are remaining, right? So fats are broken down by the lipases enzyme. I told you, remember, lipases helps in the emulsification of fat. So lipases, which is also a pancreatic enzyme that was secreted into the small intestine, they will become active and they will break down the fat with the help of bile into a di and monosaccharide. So the complex fats will be broken down by the lipases enzyme that are secreted by the pancreas along with the bile that are secreted by the liver into the intestine. Both these lipases and the bile will function together in the intestine and it will break the fats into simpler forms, that is diglycerides and monoglycerides. Now fats also got broken. So fats that was there in the food, in the small intestine, in the presence of the lipases enzyme, along with the bile, they get broken down into glycerides and these diglycerides will get broken down into monoglycerides. Now, proteins got carbohydrate, partial digestion occurred wherein the carbohydrate had already got broken down into maltose. So, remaining maltose should now be broken down into simpler forms in the intestine. We have not touched that part still. We finished studying about, that is the proteins, peptones and the proteases wherein they will get broken down into the dipeptides. So that we studied. And also fats also, digestion is complete wherein the fats in the presence of lipases and bile got broken down into diglycerides and the diglycerides got broken down into simpler monoglycerides which can be absorbed by the body. So next we'll move on to the one more enzyme which is nucleases. What this nucleases will do? So nucleases is also one such inactive enzyme that is secreted by the pancreatic juice. So the nucleases will act on the nucleic acids to form nucleotides and nucleosides. Nucleotides and nucleosides are very important because they are the main components of the DNA molecule, right? So if the nitrogenous bases has to be formed properly in the DNA and the DNA has to function well, then nucleotides, nucleosides should be produced. So what is the function? So from where it is derived? It is derived from the food itself. So the nucleases that is present in the pancreatic juice, so the nucleic acids that are there in the presence of nucleases. So obviously we would have taken in nucleic acid, right? Whatever plant material we take, in that also nucleic acids will be present, right? The DNA will have its nucleic acids. So those nucleic acids in the presence of the nucleases will be broken down into nucleotides and into simpler forms, nucleosides. So that is the function of the nucleases enzyme. So next, talking about what is the function of the other enzymes that are present in the succus entericus. What is present in the succus entericus? I told you, remember, maltase, right? What happens? We shall see in detail. Maltase, lactase, then sucrase, lipases, different enzymes are present. What each of them will do in the small intestine, we shall see. So the enzymes that are present in the succus entericus, they act on the end product. Proteins got completely digested. Nucleic acids got completely digested, fats got completely digested. Remaining is carbohydrates. What will happen to the carbohydrates? Because carbohydrate had only got partially digested and carbohydrate digestion did not take place in the stomach. Only it had begun in the mouth, but in the stomach it had not occurred. So once it enters into the intestine, what happens? We shall see. So the succus entericus acts on the end product which we had studied and they get converted into simple absorbable forms. So these are the final steps in the digestion. What happens? We shall see. So the dipeptides, from where did we get the dipeptides? Proteins, proteases, they got broken down into dipeptides, right? They got broken down into dipeptides. Now these dipeptides, in the presence of an enzyme called dipeptidases, they get broken down into amino acids. And these amino acids are absorbed by a body. Next, maltose. Maltose, it is a disaccharide, that is carbohydrate got partially digested in the mouth in the presence of salivary amylase, got converted into a disaccharide, maltose. That maltose, again, it should be broken down into a monosaccharide or simple form, right? In the presence of the maltase enzyme, which is maltase is present in the succus entericus. So it gets broken down into two molecules of glucose. Next is lactose. Lactose, milk. If we take in milk, milk contains lactose. That should be broken down, right? So that in the presence of the enzyme lactase, it gets broken down into simple forms of carbohydrates, that is glucose and galactose. Next is sucrose, sugars that we take in. That in the presence of sucrase gets broken down into simpler forms, glucose and fructose. Therefore, a body can easily absorb glucose and fructose. Next is nucleotides. We have already studied about this. Nucleotides in the presence of nucleotidases enzyme gets broken down into nucleosides. Then in the presence of nucleotidases enzyme gets broken down into sugars and bases. That is the nitrogenous bases. So this is how they get broken down. So next is diglycerides and monoglycerides. In the presence of lipases, the fats that are present in the food got broken down into diglycerides and monoglycerides. Now these diglycerides and monoglycerides should be further broken down. How it will get further broken down? Again, the lipases will act on them and break them into fatty acids and glycerol. Now in the small intestine, complete digestion of the food has occurred. These food, what will happen to it? We shall see. So the breakdown of biomacromolecules. What are the different biomacromolecules got broken down? The carbohydrates, the proteins and the fats, right? So much got broken down. So the breakdown of biomacromolecules mentioned above, whatever I had told you, occurred in the duodenum region of the small intestine. All these processes occurs in the duodenum region of the small intestine. And the simple substances thus formed are absorbed in the jejunum. So we had studying about the structure of the small intestine. The small intestine are made up of three parts: duodenum, jejunum and ileum, right? In the duodenum, all these breaking down of the foods into simpler forms will occur. After that breaking down of the foods, then the simple substances will enter into the jejunum and the ileum. And whatever undigested and unabsorbed substances are there, it will be passed into the large intestine. So I hope you understood. So whatever food got digested will enter into the jejunum and ileum part of the small intestine, and whatever undigested food is there, it will move into the large intestine, because the large intestine is the one that is connected to the anus through which the undigested food gets excreted out, right? So that is why. So this was about the end products, whatever is formed. So next, the food, the undigested food has entered into the large intestine. We shall see what happens in the large intestine. So here, no digestive activity occurs in the large intestine. So what are the functions of the large intestine? So the functions are, it absorbs some water, minerals and certain drugs. So that is the function. Next is, it helps in secretion of mucus, which helps in adhering the waste, that is the undigested particle. It helps in keeping the undigested particle moist, so that the undigested particle can neatly pass through the large intestine and leave the large intestine. The undigested unabsorbed substances, which is called as fecals, then it will enter into the cecum of the large intestine. From the cecum, it will enter in through the ileocecal valve, and from the ileocecal valve, it will enter into the anus. Why is this, or the rectum? Why is this ileocecal valve present? The ileocecal valve is present between the cecum of the large intestine and the rectum, because the undigested food or the excretory waste should not go back again to the large intestine, should not revert. So that is why. So next it will enter into the rectum, wherein the defecation process will occur. So it will be stored in the rectum until the defecation or the excretion process occurs. This is the function of the large intestine. So large intestine will never take part in the digestion. All the digestion would have been completed in the small intestine itself. So this is about the digestion process. So next, let's talk about the activities of the gastrointestinal tract. So the activities of the gastrointestinal tract are always under the neural and the hormonal control. They are involuntary in nature, right? We cannot control them. They are involuntary. They are controlled by the nervous system and various hormones that actually function together to keep a proper coordination between the functions of the process of the digestion. So whatever sight, smell or the presence of food in the oral cavity can stimulate the secretion of saliva. So how will our mouth or the salivary gland understand that the saliva has to be secreted? So as soon as we take food into our mouth, we will start chewing, right? Why? Our brains will receive signals through nerves saying that the chewing process has to occur. Next, as and when we chew, the saliva gets secreted. Why? Again, the brain will receive the signals through the nervous system that the saliva has to be secreted. And the gastric and the intestinal secretions are also similarly controlled by the neural signals. So all the secretions so far that we had studied, all are controlled by the neural signals and also certain hormones. So this is the activities of the gastrointestinal tract control. So they are controlled by the neural signals and the hormones. And also the muscular activities of different parts of the alimentary canal are also controlled, and not just that. So the alimentary canal, they can also be controlled by the neural mechanism, both by the local and through the central nervous system. So the central nervous system also activates the muscular movement. So muscular movements for the movement of food through the esophagus, there also that muscular movement was required. In the stomach also, for the churning process, that muscular movement was required. In the intestine also it is required. So for all that process is controlled by the neural mechanism, be it the local neurons that are there, or the central nervous system. So, and hormones. So, I told you hormones also control the gastrointestinal activity. So, the hormone controls the secretion of various digestive juices, which is carried out by various local hormones produced by the gastric and the intestinal mucosa. So, gastric juice secretion also, or the various secretions that are there, enzymes that are secreted, all those are controlled by the hormones that are secreted by the gastric glands and the intestinal mucosa. So this is how the activities of the gastrointestinal tract are controlled by the nervous system as well as the hormones. So this was about this particular session wherein we completely understood how exactly the digestion occurs right from our mouth up till it reaches the small intestine. So digestion begins in the mouth, wherein the carbohydrate digestion occurs in the mouth, and the complete digestion of the carbohydrates, fats and the proteins occurs in the small intestine. Then the undigested food will enter into the large intestine. Large intestine will never participate in any of the digestion process. Its only function is to eliminate the waste material. So that we had studied. In the next coming session, that is the last session of this chapter, we shall study about the various disorders that are there in the digestive system. So I hope you understood this session well. We shall meet again in the coming session. Thank you.